go. Take a look. You guys have pictures on the front. All right, again, you know who this is brought to you by. Now, we are going to count the number of cells and identify them as one of the stages of mitosis or the one that is interphase. So it probably helps to have this picture here. Now, we have your different areas. So you got a slide that looks like this. It says view 1, that's area 1. On the back is view 2. That's what you're going to write your numbers here, your raw numbers. Okay? Then, view 3. Okay? So we're going to try and do this. We're going to pause a lot. When you're done, you got to total this, all right? And I can give you an idea. This is from previous years. As you look, you'll see this is the total number interphase for the first, second, third view, even though it says area. And then you're going to have to total it. All right? So I'm going to get right to counting, and then we'll come back once we get some results and talk about what's going on. Now, here's the logistics of how we do this. It probably helps if you have another paper, say, to kind of help yourself count. I don't want you writing on this with expo marker. It just makes too much of a mess. So if you look at right here, that's row 1, 2, 3. If you go this way, it works. The big question is, which ones do you count that are on the periphery? And we're going to have a general rule of thumb. If the whole nucleus is in, we'll count it. So are we going to count this one? No. No. All right, we're not going to count that one. We're not going to count this one, but we'll go ahead and accept this one. And again, when you're counting, if you want to use a scratch sheet of paper, those who are used to doing tallies, well, I want the actual numbers here, so do that on a scratch sheet of paper. So let's go ahead and get your data. We'll talk about how you're going to do this. Follow this calculation, and ultimately we're going to put this on the back with the pie chart. All right, so our objective is to get this page done. We're going to skip this, go right from this to the back part. Okay? So I'm going to pause until, and just go ahead and leave view 1 up there. And I actually took a picture, so I'll probably put that up a little bit later on. So let's reconvene in a couple minutes. All right, I've been asked this question by a couple of you, so let's see. I'm going to go ahead and point. Tell me, this first row, what is this? Follow the mouse. Interphase. What about this? Prophase. How do you know it's prophase? All right, 2 ways. You can see this nuclear membrane starting to disappear, and right? You can see the chromatids. You can see chromosomes. Remember our mantra. If you can see chromosomes, it's mitosis. If you can't see chromosomes, it's interphase. Like this picture over here. All right? You can't see chromosomes, so that's chromatin. Versus, you can see chromosomes. They're sister chromatids because they're doubled chromosomes. We called that dyad earlier. And you can see the disappearing nuclear membrane. All right, so something else. What about this? Metaphase. They're lined up across the middle. You don't really see anything else. All right, what about over here? Anaphase. And then let's find, all right, so let's count. 1, 2, 3, 4. 1, 2, 3, 4, 5, 6. Come all the way over here. Take a look at this one. Telophase. One way to know, look at the size of this cell. See that? Cell next to it is like a parent cell. It's going to go through division soon. That's interphase, but it's half the cytoplasm. After cytokinesis, the cell has half the it has half the cytoplasm. It's got 100% of the genetic material because it's a clone. So that's one of the ways to kind of look through with this. So, and you can see here's a prophase where the nuclear membrane is starting to disappear. But you see the nuclear membrane starting to appear. All right, we'll talk about the calculations next. All right, you guys, watch so after you get your data, let me just kind of show you, hope that's in the view here. You got to add these 3 together. That's how I got that number. And same thing. Then all these get added for the total. Now that's how you're going to figure out the minutes. Okay? What does it say? It says the total number in each stage divided by the total number. So you take 180, and in order to get that number, I take 185 divided by 233 in my case. Now again, your numbers might be slightly different. That's going to give me a decimal, right? Then I got to multiply that by 720, and that's going to give me 571.6. I'm going to round up to 572. So that's how I got this number. You got to take this number divided by your total, then multiply by 720. Okay? Now the next thing we're gonna do is we're gonna talk about the back part. All right. Now, how do you get from here to the pie chart? First, let's recap. All right, guys, listen up for a second. We got these. This is the total count from the interphase, from prophase. This number is added together to give you this, and in order to get the percent, you take like in this case, I took 185 divided by 233. That's how I got, and then I multiplied it by 720. Following this, that's how I got my 572. Now, what you need to do is take all of these numbers and divide by 18. Why? Okay. In order to figure out how to do the pie chart, okay. Pause for a second over there. In order to figure out the pie chart, you're going to take that number. You're going to take each one of these numbers and divide by 18 and write it right here. Now, you guys, some of you started coloring it in. You're going to pick a color for each one. That number, if you look at what I have here, and we're going to close up this video, that number tells me how many of these I'm going to shade in. So you have a color code. So let's go ahead and I'm going to go and finish up with this. If anyone's absent, you're watching this video, you're expected to do the lab by copying down this data. And this is what the pie chart will look like after you've done all this. Something like that. So guys, take a quick look. Does everyone see? You'll notice interphase is the most numerous stage of the cell cycle. All right, that's our lab.